let's get started. There's 2, 4, 6, 8, 10, 11, 7 people missing. People are starting to skip already. What did I want to say? I guess the recordings don't really help in that perspective. I used to teach genetics, and after like 3 or 4 lectures, because I was recording, half the class was missing, you know. In any case, that's okay. So today we are going to start talking about how to get the human genome. So we'll start by talking about the project that, the 2 projects that were main in getting the human genome, and then we'll, dissect them step by step until we get to the human genome. We'll do something similar to the yeast genome. Only difference is that now this topic will probably last for like 4 lectures, okay? 4 or 5 lectures. That's how long it is. So that's that. I just wanted to say that today is the day where I assigned to you the paper that you have to read and write about. So it has to be submitted, the lecture, right before the reading week. I put here the grading scheme so you can read this to understand what's happening in here. And obviously, if you don't get something in here, or if you're not too sure about how to approach this problem, you can contact me and we'll talk about that, okay? So the paper is about cancer. So we'll be looking at genomics in the context of prostate cancer. You guys will write about this, and then I will talk about this, okay? What's expected is about 5 pages long, the essay. Obviously, we can get into what's the font size and all that, but I think you guys are used to this, the 12 pixel and Times New Roman and all that stuff. Just make sure that your writing style is scientific. This is not like the homeworks where you were just, giving your opinion. In this case, it needs to be scientific. That means your references must be peer-reviewed. Don't just pick up news articles and then talk about that. In this paper, so there's a lot of data. You'll probably not understand a lot of it. If you're taking functional genomics, probably you will do. So I highly suggest that if you don't understand something, that you go read on it first and then come back to here. And then obviously you can use those references to support your claims and all that, okay? The idea is to describe the paper in your own words. That's the first part. So you'll give a brief, so let me just bring this up, where is it? Okay, so you're expected to talk about the big picture, position it within the field, explain the knowledge that you need to understand this kind of paper. Very briefly, because each technique you can write a paper on it, and actually they do exist, these one technique 10 pages paper, so that's not what's expected here. In the methodology, you have to talk about the methods used, the statistical tests used. Are they relevant? Not. Why aren't they relevant? Why are they relevant? And obviously show that you understand the experimental design. So everything is written here. Results, describe all the results. And also, as I was saying, the opposite side, so the limitations. What is the limitation in this paper? What are the limitations of the experiments? Let's say you find an experiment that could have been done better or whatever. I'll let you read all this, okay? Overall style, don't base yourself on what ChatGPT is going to push out. Make sure that you take a look at this, because sometimes the style is not what is expected. So make sure that after you put it in there and do whatever you have to do with it, that it comes out as something scientific and not just, you think that it's correct, but it's not correct in terms of style and language use, okay? What else? What else? Do you have any questions first? No? Simple? Okay. This is an exercise to get you to know how the reviewing process happens and to get you to think on your own. So this way you can get the skills to read papers. You guys probably already read a few papers since the beginning of your studies. Now you get to review them. And I think you have an assignment similar to this in functional genomics, right? Anybody? No? Nothing like this? Okay. So the techniques that you learned, you'll be talking about them here. in functional genomics. That's probably the same ones. Okay. Cool. You have 1 month to do this. I don't think you're going to be late. But let's just get the late penalty explained right away. So I'm not the type to give 0 right when you miss it. So if you submit it late, what I'd like to do and what I'd like other profs doing is remove 10% per day. Okay. But I think if after a month and all the tools you have, you're still late, there's another problem, okay, that we have to take a look at. So this paper, I'll present it completely in the lectures. I'm not sure if I'll do it. So this paper will probably take like 2 lectures to present. So I think I'm going to do it quickly in 1 lecture, remove a lot of the extra stuff so that we can cover other interesting topics, okay? Instead of just focusing on prostate cancer and Neanderthal, we'll also take a look at gene therapy and I forgot what I wrote for the other one. Actually, I can take a look here. What was it? The microbiome. So genomics for the microbiome. Cool. The final exam has been, the tentative schedule came out. I don't know if you saw it. If you didn't see it, the exam is on the 5th. That's a Thursday. The 5th, it's a Thursday at 2 p.m. The rooms are not yet assigned, so I will let you know about that. Okay. So in this exam, we didn't even do the midterm. We're talking about this exam, but it will be exactly the same as the midterm. So just bring a pencil or many pencils. Make sure you also have an eraser and all that stuff, so that you can answer the written questions. Okay. And that's pretty much it for that. Any questions? Not everyone has submitted the assignment yet. You have until tonight, 11:59, I think. Is it because you're just leaving it at the last minute, or there's issues? Anybody? Or you already have too much assignments and you're trying to catch up? Sorry? I just want to check it before putting it in. One last time? Yeah. I think Sarah was the first one. I was like, okay, first day. So the pressure is on her now. It's okay. Anyways, you have until tonight to do it. Should be quite easy, but sometimes we let the work take the time it takes. In any case, so human genome. So we got the yeast genome. Now we're trying to figure out what the hell does the human genome look like, because most scientists were not actually interested in the yeast genome. We don't really care about it. We care about humans. And so they did it. They sequenced the yeast genome just to acquire the expertise to be able to do the human genome. Some of them obviously did it because they wanted the sequence of the yeast genome, but most of it was directed towards the benefit of humans. Okay, and so when it was sequenced, we realized after doing some functional studies, there's about 20 ORFs, 20 genes that are in the human genome. But the problem is that the sequence was about 100 times longer than the yeast. And so that was kind of weird. So that's where we understood that there's other pieces that are not found in the yeast, or at least found in limited numbers. And so because the yeast has about 6,000, let's say 5 something confidently assigned ORFs, then it appears to be 4 times more complex than the human genome, because this one has about 20,000. Obviously, I didn't want to do the division here and say 3 point whatever. And so what happens here is that that's not true. It's not 4 times more complex. It just has about 4 times more genes, but in reality is much more complex because there's a lot of DNA sequence that we don't know what it does. Okay, so we'll take a look at how we were able to go from not knowing what the genome looked like with some experience in sequencing the yeast genome to figuring out the human genome. And this is the slide that I just said. So we'll go to the next part. If you remember from the first lecture, I talked about the Human Genome Project. This is a US-funded project that started in 1990. And the goal was to sequence the human genome, right? So it was a team of about 2,000 people, if I remember correctly. And they were aiming to get it by 2005. So that's about 15 years of work, okay? At the same time, there was a private company called Celera Genomics, which was created by Craig Venter. He's pretty well known in the in the biology, synthetic biology community that was saying that they will get it in 2001. And the Human Genome Project was run by Francis Collins, and this is Craig Venter. So these guys were fighting. They were trying to get it at different times, and then what happened is that, you know, this always happens in science. So you have two people that are competing for the same thing, and then so one scoops the other. These guys, luckily, they ended up working together, and they finished the sequence of the human genome in 2000, released it 2001. Okay. These are the papers that came out of this. So you can see, you can read about them. Just search this on Google, and you'll find them. You can see what the hell they did and how they got that. Another paper that came up, because the human genome was complete, but it was a draft, so you had to fill in the gaps and all that. So this is another paper that came out right after to fix some issues that existed with the initial assembly of the human genome. Okay. Cool. So let's see. So there's 5 steps where scientists went from 1 to 5 to get the actual genome. The first one, you have to characterize the genome. You have to understand what it looks like. Yeast has 16 chromosomes, but the human, we don't know how many it has. You have to understand the shapes of these chromosomes, things like this. You have to construct the libraries, so same thing as yeast. Map them using contig assembly, as we saw in the yeast example. Sequence the contigs, and then annotate them, right? And this is the hardest part. Annotation requires a lot of functional studies, so you'll talk a lot about annotation in your functional genomics class if you're taking it. If not, I'll be briefly going over it, but it's the idea of assigning function or function to a gene based on sequence. Okay, and so one thing that is important to understand here, the difference between the Human Genome Project and Craig Venter's approach is that Craig Venter just went straight to sequencing. He did not care about these contig assembly whatsoever. So break the DNA, sequence the pieces directly instead of building this library. Okay, so this is the example here. The HGP approach, Human Genome Project, is called also hierarchical shotgun sequencing. And so what you do is that you break it into pieces, and then you build these contigs that you will sequence separately, and then you will try to assemble the genome based on the sequences of these and the overlapping sections. Okay. Celera's approach, which is now kind of the way that we do it, is that you break it into small pieces. And you go directly to sequencing those pieces in one single, let's say, test tube. Okay. This works in a similar way as this. The only difference is that you're basically using these adapters on the side, and these adapters allow you to kind of link up the pieces between each other. And obviously you need, well, at the time they were advanced computers, now it's nothing, but you need computers to be able to do all the calculations to see where the sequences go together. And even there were errors in the genome which were corrected in here. This is a 2022 article, so it's quite recent, and we're still doing corrections to the human genome. Okay. So the contrast between the two approaches. So they're not based on nothing. They're based on examples that were previously done in more simpler organisms. So the HGP project was structured like the yeast genome project, which we saw. Celera's program was looking more like the influenza bacterium sequencing, which was the first bacterium that was sequenced. Okay, and both of them are good. It's just Celera's approach is much more faster because you don't need to build all this library, look at the contigs, etc. What else? Okay, so I think this is what I just said. So you don't build the maps and contigs. You just fragment this genome and then sequence. You use computers. Okay, I think I said all this stuff. So let's go to the next step. So how do you actually characterize this genome? First step is called karyotyping. So you need to understand the content of the nucleus of that cell, right? And for that, you need to understand what chromosomes look like, how many there are, if they're in diploid state, triploid, tetraploid, whatever, because that will affect how you sequence the genome, right? And you need to also map, right? So if you have two chromosomes that are homologous, then mapping becomes more complex, right? So we'll take a look at this. You will take the chromosomes at metaphase, right? And then you will stain them to be able to see their morphology. So I'll talk about the stains here. So there's this GIMSA stain, which will produce these unique patterns based on the AT-rich regions in the chromosome. And so each chromosome has a different banding pattern, and it's unique to it. And so you can recognize the chromosomes based on that, okay? This also allows you to identify abnormalities in the chromosome. For example, if you see that this part moved to another chromosome, you know that a translocation happened. Things like this, because the banding pattern will change. For example, if it becomes 3 lines here, you know that this is not actually this chromosome. It came from another one. And so this is what it looks like. Obviously, humans are diploid, so we have 2 copies of each chromosome. Technically, we have the last 2 chromosomes, the sex-linked chromosomes, are actually unique. The others are all homologous. And so if you take a look at chromosome number 10, for example, they have the same banding pattern over here. Obviously, these are just squished because when the picture was taken, they were probably separating, and so one pulled faster than the other. But if you open this chromosome up, you'll see that it's exactly the same as this one, except for these 2, which were kind of unique, okay? So if you're trying to map the genome having this much complexity, you don't know if a gene came from here, or from here, right? Before we get into that, there was another type of staining that was created. So this staining was called FISH, fluorescent in situ hybridization, and so it allows you to fluorescently label the chromosomes. So here it's more radioactive. Again, same examples as we've seen before. This one is more fluorescent. This allows you to kind of differentiate the chromosomes in different colors, and so each color is based on a pattern that is specific to that chromosome. So then you can see the homologs together, etc. Like for the example, these 2 are homologs. These 2 are homologs. These 2 are homologs, etc. Okay. So remember, FISH uses again the same cameras that were used for the DNA sequencing using fluorophores. So it's CCD cameras, and obviously you have these fluorochromes and these filter systems that detect different lights, different wavelengths. So obviously, if you have 23 chromosomes, you'll probably paint them in 23 different fluorophores. So you need a filtering system that is capable of separating all these different colors. Okay. So this is pretty, but what is the purpose of this? So the purpose of this is that you can see translocations more easily when you're using this FISH hybridization. And so if we take a look at here, actually let me just get to this point here and then I'll come back. And you can also take a look at the structures of phylogenetically related organisms. So if organisms have similar chromosomes, you'll know that they'll have the same colors, things like this. Okay, same size, same color, etc. And so if we take a look at this, this is a cancer cell that has a translocation that happened. And so if you take a look in here, you will not see any translocation happening. But because you take a look at this FISH staining here, you can see that there's a small piece of red here that is not supposed to be there. So this is due to translocation. There's another one here, for example, that is green. You could see it technically on these guys if it was big. For example, you'd have the banding pattern. Wait, you have this banding pattern here, and then you have like a big piece that was translocated. But if, for example, the translocation happened just inside here, you will not be able to see it. because the color remains the same, and if it goes to another chromosome where the color is still like this, then you don't know if there was a translocation. While with this fish, you can actually see it because the colors change. Okay. One thing that fish does not allow you to see is inversions. And I think I talk about this a bit later. So before I get into that, this is just another example where you see a lot more translocations. And these are typical of cancer cells. So you can see them here, here, here. And you'll see why this is important. It's because we need to understand the structure of the chromosome so that we can understand what's in it, right? If we cannot separate them and distinguish them, then we will not know that that gene came from this chromosome or this one or this one, okay? So you're able to detect these abnormalities in the chromosome using these two methods. The problem is that when you're using fish, you cannot see inversions. The reason for that is because inversions will basically just lead to, for example, an inversion happening between this and this. Oh no, not inversion inside the chromosome. And so you will not know that this piece here, for example, moved to here because they are the same color. While if you're looking at the previous method, the GIMSA method, an inversion will likely disrupt the pattern in here. If it goes from here and then ends up in, let's say, this black region here, then you'll know that there was an inversion there. So these are two different methods that are used to understand the chromosomal set of humans. Okay. Let's take a look at an example where these stainings are important to figure out phylogenetically related species. So here we're looking at the deer, called the muntjac in scientific language, let's say. So the Indian muntjac and the Chinese muntjac are related species. The only difference, if you take a look at this here, it seems like this one has 3 chromosomes. So one yellow, one red, one green, and obviously it's a diploid, while this one seems to have a crazy amount of chromosomes. And so if you stain them both using the same stain, you get this pattern here, right? So do you guys have an idea on how these two are related if they don't even have the same number of chromosomes? What's one reason? Yeah?
The chromosome could have just been fragmented.
Fragmented, Joey, same thing?
Yeah, I was going to say that, but then I thought of something, but then I never.
Okay, so then if so most people would think that the chromosomes actually fragmented, right? It seems like this one here was broken in multiple pieces, and then there were obviously some translocations that happened, as you can see. Reality is, if you take a look at this paper studied them, it's the opposite that happened. So all these small pieces here went together, formed these 3 chromosomes, okay? And you can study this, so I didn't read this paper, just know that it exists. But there's probably a scientific way on how they went from which one happened first, you know? Is it fragmentation, or they went all together to form these chromosomes? So you can take a look at this to see how fish is used for that. So this is just the 2 pictures that show how the different parts come together to form each of the chromosome. So this is fusion of the chromosome number 3. From the Chinese muntjac to the Indian muntjac. Okay, and they did this for all the chromosomes. This I just said. Karyotype is important because it provides the number of chromosomes and the ploidy, and this affects how you sequence a genome. If you have a haploid set, for example, in the gametes, or if you're looking at a haploid set, for example, in yeast, then it's much more easier to do mapping compared to if you have a diploid, for example, your somatic cells, epithelial or whatever. And then if you're taking a look at plants, this is even worse because they're polyploid, they could be tetraploid, triploid, tetraploid, etc. Okay? You need to be able to separate the chromosomes to find just the unique ones and then go on from there. So our goal is to build a map of the human genome. So again, there are 2 ways to do this. There's the genetic map, which you learned in genetics first year probably of biology. And then there's the physical map. The genetic map, as I mentioned earlier, it's based on the inheritance of genetic markers. So you can compare how two markers are being inherited in a relationship to each other, and that will tell you how far they are from each other. So this is, it takes a look at the recombination frequency, and then you can use that to calculate the distance between two genes in centimorgans. Okay. This was very useful because in yeast, since yeast can be haploid, then you can study this much more easily, and it was used on yeast chromosomes to figure out the pattern of genes. 1 centimorgan gives you about 3,000 base pairs, so we don't know this yet at this level. Actually, for yeast, we did, and this is just based on how two genes are recombining together. So if you have two genes that affect two different traits, go together 80% of the time, that means they're super close. Okay? If they go at 50%, probably they're on different chromosomes, things like this. The physical map also uses these markers, but these markers are physical. So they're stretches of sequence that is kind of unique that you use to build this map. And karyotyping is one example on how you can build this physical map, although it's very low resolution. So you're creating here, should I put this picture everywhere? Technically a physical map, because these are specific sequences that are recognized by the GIMSA staining. It's not random. Okay? So you have somewhat of a physical map, but obviously you need to improve on this to be able to sequence the human genome. Before we get there, genetic maps are very hard to do for humans. We're not like yeast or Drosophila or things like this. The reason for that is that you cannot really do test cross in human. You can just take one person that has a disease, tell them go make a baby with the other person that has a disease, and then see the kids, you know. It's a little bit unethical, right? Instead, what you do, you kind of need to find people that have the disease, and then study those people if they're willing. That's the first part. And then obviously, for very rare diseases, it's very hard to find. And for it to be statistically relevant, you need to find a lot of people. Okay. For example, the only trait that could be studied were the ones on the sex-linked chromosome, the X chromosome, for example, because the X chromosome was unique. So either you have one copy of it or two copies, and so in that case, you can start mapping based on that. But for the autosomes, you have 44 of them, and you don't know if it's chromosome 10 that's giving that gene, or chromosome 8, or the other homolog, or whatever. Okay, so the only one that you could use classic genetic mapping on is the X chromosome. That's the first part. And a good example of this is red-green color blindness. Here you can't distinguish these two colors. So in class, in genetics, I used to give them like the example that the optometrist does, but then I realized that some student might not feel comfortable doing the test, so I removed it. But red-green color blindness, I had one student, and usually it's linked to hemophilia, right? But how do you figure this out? They're both on the same chromosome. So for this, to figure it out in humans, in yeast, what you would do, you would just take one that has hemophilia, one that has red-green color blindness, just crazy example, made them, and then see if they become, you know, both at the same time, right? In humans, you need to have the pedigrees, right? Because you cannot really take one person that has red color blindness and the other one that has hemophilia and tell them, make babies, I want to see what your babies have, you know? It's actually very unethical. So what you do is you find these people, and then you study their pedigree to see if both traits occur together. And obviously, they do occur together. So basically, the distance between them is less than 50 centimorgan. And so that means that they're very close to each other on the chromosome. So the genes that do this, okay? And this segregation is very rare. So again, just showing that it's actually almost impossible to use classical genetic mapping for humans. You can read on it on this paper here. Not only that, there's a lot more issues with the genetic mapping. One of the issues is, obviously, as I said, you can't direct mating, like for model organisms. You have sometimes to pool results together. So this happens because you can't find enough people, and so you will pool different studies together. So this creates already its issue in terms of inconsistency and reproducibility. And sometimes diseases happen due to different genes. So you can have 10 different genes giving you the same, so a mutation in 10 different genes could give you the same disease. For example, hemophilia, there's 2 different mutations that give you hemophilia, and color blindness, one of them, and so you can see the different patterns that can occur, and then you have to combine these 2. If you combine these 2 groups, you'll end up not knowing if it's the same gene that you're comparing, or if it's 2 genes that give the same disease, so the same trait, and then to map that, it's almost impossible. Okay? Next question. How the hell do we map the genes to autosomes? So for the non-sex linked, you need some differences between the chromosomes. So if you have homologs, you need differences between them, and then obviously the autosomes need to be different from each other. One good example is the HLA locus for the blood groups, ABO also. And so this one is easy to work with because you have multiple, you have polymorphisms that occur in here, and then you can either be blood type A, B, or O, and then AB, obviously. And so this one here is easier to map, kind of, okay? But there are others that are quite complicated, and so you need different methods to do that, okay? Questions? Easy? Sure? Better see this on the exam, okay? So it was very hard to use classical mapping strategies to actually map genes in the human genome. So you needed to find traits that were linked to chromosomal abnormalities. And these abnormalities need to be kind of like translocations or inversions or things like this, so that they're big enough to actually used to map the genome. And so one example is the Philadelphia chromosome, which was a translocation that was causing cancer. And so you can kind of figure out where the gene that's causing this cancer, the gene that's causing this cancer is. You can Google Philadelphia chromosome, you'll see what it is. But the problem is that visible chromosomal abnormalities affect a lot of genes, and so another issue. Solution is coming soon, okay? So there was one breakthrough that helped in this, and this is probably something that you learned about in the past. So along your DNA, you have these restriction enzyme cut sites, and so these are different because they are polymorphic. So some people have the same pattern, others don't. Well, you don't have exactly the same pattern, and so if you start cutting this DNA using restriction enzymes, you will produce different fragments. And you can then, for example, let's say a gene has a mutation in it that causes, for example, I don't know, problem with your, let's say, sickle cell anemia, okay? You have a problem in the gene that's important for that, and then let's say that mutation affects the restriction enzyme cut site, then you'd be able to say that this person has a fragment that's this big, and this one, because they have the disease, they have 2 fragments of this size. And so you can kind of map where the gene for hemophilia, for sickle cell anemia is, okay? Obviously, this takes a lot of work, especially for 3 billion base pairs. So it's not the best way to do it, but the first example of restriction fragment length polymorphism mapping was done in yeast by Davies, Ron Davies. You can Google it up, and you can see his work, okay? Okay, so what you can do, though, this is the solution to all the problems that I just created and presented. So for genetic mapping, what you do is that you'll use these DNA markers that are polymorphic. So basically that more forms exist than just one. Like, for example, restriction fragment length polymorphism, microsatellites, which are just small repeats of the same DNA, single nucleotide polymorphisms. So these are just changes within the DNA that are different from person to person at a single nucleotide locus. And you can use these to map genes between them. That's in terms of how they recombine and whatnot. If you're trying to build a physical map, then they don't need to be polymorphic, because now you're dealing directly with the sequence. And so because you're dealing with the sequence, it's much more specific. So you're not going approximately. Instead, you're using a sequence that is found in the DNA at a specific locus and not on another chromosome, let's say. Okay? And this is done by sequence tagged sites, and we'll take a look at that. Jeez, going super fast. Let's see. So there is these. So one of the way that this was done for the physical mapping is through these hypervariable mini satellites. No, not physical mapping, but genetic mapping. And these hypervariable mini satellites are basically, so the mini satellites or microsatellites are small pieces of DNA that just repeat, right? And so the idea is that there's many repeats of this sequence, but they're different between each chromosome and each person, okay? So for example, you could have 2 repeats, him 3, etc. Okay? And so they're spread out throughout the chromosomes, and they're polymorphic because they are highly variable. Some chromosomes have 5, others 10, etc. And so this is just an example. There's actually thousands of them. And so what you do is that you can do DNA fingerprinting and take a look at the different patterns of these microsatellites, and then identify the genes based on that. Okay? Because then what you would do is you take, I think I'm explaining it in the next one. Okay, I'll give you an example here, okay? So let's take a look at how you can detect these microsatellites and then map genes from there. So you can imagine that, let's say there's the mother and father, and they all have different alleles of the same gene. And so obviously this is a diploid. So in this case, you have 4 different repeats, 4 similar repeats, so they're identical, right? And for this allele, you have 2, 4, 6. Here you have 2, and this one, I think, 8 or 10, okay? When you're trying to figure out when you're trying to map this, what you can use is primers that flank these microsatellites that are known. And then what you do is that you'll run them through PCR, and it will give you different fragments of different lengths. And then you run them through the gel, right? And you will see the banding pattern of each of the individuals. And then from there, you can figure out which allele gives you the disease, and you can use that to map along the chromosome. Okay? For example, here, if you take a look at here, so let's say the mother, right here, the mother has the disease because of the black circle. This mother has the allele number 1 and 2, and so if you produce these PCR fragments, you'll end up with these 2 bands, which are in the middle. If you look at the father, which does not have the disease, then you end up with these 2 bands, the biggest one and the smallest one. And then if they have kids, then you have a specific banding pattern. And depending on the banding pattern, you can try to map a certain trait to a certain locus, okay? For example, here, the first offspring, or the first daughter, has allele number 2 and allele number 3 from, allele number 4 from the father, okay? Are you able to take a look at this and figure out which allele gives you the disease? Let's do a little pop quiz. Yeah. Sorry? Number 2. Yeah. Yeah, number 2 is the allele that gives you the disease, because you can find it here, here, and here, but not in the healthy ones, and so it's allele number 2. Yeah?
Are like the sequence of DNA, is it the macrosatellite that is responsible for giving the illness, or is it like this is the gene? Where does the macrosatellite
come into play? So you need to Okay, I see. So you're using the microsatellites as a genetic marker in relationship with the gene that gives the disease, right? So these microsatellites happen along the chromosome everywhere at different distances. Some people have 10, let's say at this space here they have 10, others have only 2. And so you can use these in relationship with the trait that you're looking at, and based on how you cut your DNA, you will see a different banding pattern, okay? And then because of that, then you know that it's positioned here, depending on the size that comes out, or if it's further or not, okay? You're actually looking at these guys here, right, in relationship. So this is just a genetic marker that you're comparing to. Questions? No. You can't do like for yeast where you're looking at, let's say, auxotrophy, which basically they need to grow on this or that. In this case, the markers that you have to use are not supposed to be disease-causing markers. They need to be kind of these neutral markers that then you can take a look at, because you can't really take a let's say, for example, you're using as a marker for humans the gene that causes hemophilia. Then you can't just get all the people that have hemophilia and then sequence and see in relationship to there. So what you do is that you find these neutral sequences in the DNA that are variable between each of the chromosome, and then you use those as markers to map. Okay? In yeast, you'd say, okay, this gene here causes forces the yeast to be supplemented with, I don't know, histidine, okay? And then you use that and see if, for example, another gene that has another requirement, for example, resistance to whatever antibiotic or whatever molecule, and then you say, does it grow only on this histidine? Does it grow without histidine? And does it grow with histidine and is resistant to that? And if you know that it's resistant to antibiotic or whatever and also requires histidine, then you know that those genes are close to each other and far from each other. But for humans, cannot do that. So you need these microsatellites, okay? So the microsatellites are 2 to 4 nucleotide units. For example, here, you have cytosine and adenine, and then just repeated, etc. So there's different combinations. They are dispersed in the genome, highly variable depending on the person you're dealing with, but also depending on the chromosomes themselves. The DNA that's surrounding the repeat can be amplified by PCR, and this is important for producing these gels. And they are used as molecular markers. So as I just explained, these markers are important to determine positioning against other genes. And then they're used a lot in DNA fingerprint analysis. So we were talking in the first lecture about how the police figures out if you're a suspect, if you're a criminal or not. Before we get into that, I'll just show you an example of the microsatellite marker map. So in this case here, this is all the chromosomes for the human. I think there's the Y missing that I just realized. Maybe I cut the picture. It's actually a very good question. I have to figure out why the Y is missing. So I'll take a look at this. But this is the banding pattern for these microsatellites along the chromosome. And so obviously, each of them is different. And so you can kind of take this one, for example, and map in relationship to this one here, the other genes, okay? So if, for example, you see that it's always, so let's say, for example, a gene that is close here is always inherited along with a gene from here, that you know that they're probably on different chromosomes, they're on the same chromosome. If they, let me repeat that. If a gene is right here and is inherited all the time with this one, that means probably they're on the, let me forget that, okay? Because I have to think about this myself. But basically, just think of it as, if you're using this unique set here, then you can just figure out if the genes are on the same chromosome or not. The mistake that I just made, it's because you need to separate the chromosomes, each one alone, and take a look at the fragments they produce. And we'll take a look at this in like 2-3 slides, okay? So forget what I just, blabbered about. This is the paper, the first paper that showed the microsatellite map, and you can take a look at them, okay? Now I'll explain this stuff here that I just, mixed up in a second. Police. So the police work. So this is the question that I got in the beginning of the semester. How do the police figure out if you're the criminal or not? So first of all, they'll get the suspects, and those suspects will have different banding pattern, different numbers of these short tandem repeats. So for example, this one here will have, let's just take an example. This one has 5 and this one has 3 here. So then when you actually run the PCR reaction using these primers, you'll have, and then you run them through the gel, you'll end up with different banding patterns, right? And then you can obviously compare it to the crime scene and see that suspect 1, actually none of them is, no, suspect, yeah, this is supposed to be suspect 2, okay? This is number 2, okay? So these 2 are the same, so this one here is probably the criminal. Actually, it is the criminal because it's highly specific. And the FBI uses a combination of 13 short tandem repeats. So this is one of the questions that came up. They also use single nucleotide polymorphisms depending on which agency you're dealing with. But the FBI uses 13 of them. And they're called the 13 CODIS core STR loci, short tandem repeat loci. You can Google this weird sentence here, and you'll see how the FBI uses these. Okay, this is just their names, so you can see where they are found and on which chromosomes. So it's all these guys here that are used. So every person has a different combination of these. Different number of repeats here compared to, and then if you combine them all together, you can imagine that you create billions of different combinations. Make sense? Okay, so that was for genetic mapping. So just figuring out how genes are linked to each other based on their recombination frequency. What we're interested in for the human genome is more the physical map. And so for the physical map, what you need is a sequence that is unique. And so what you'll do is that what you're looking for is what's called a sequence tagged site, a short sequence of DNA that is less than 500 nucleotides and that is found only once in the genome. Because it is found only once in the genome, that means it's specific to a chromosome. And this allows you to differentiate between the chromosomes and do physical mapping. So you could generate, so these sequence tagged sites can be generated and then they can be detected using PCR. And this is critical to actually figure out the pattern of genes among a chromosome in terms of their physical location, so how far they are from each other. So the experiment that was done to do this was using a mix of human and rodent cells. And the way you do this is that you'll take a cell that has a rodent human cell with one of the human chromosomes. So you can separate chromosomes using gel electrophoresis. And then you will basically give the rodent human cell a lethal dose of radiation. Radiation, what it does is that it induces chromosome fragmentation. If you're aware of, so cancer, one of the treatments that is used is radiation. So obviously, first you try to do specific drugs for cancer, but sometimes you need to use a different approach when you're not able to treat it, so you will use radiation. Chromosome fragmentation itself can cause cancer, so you can see how the treatment itself could make it worse. So this is why when you're dealing with, for example, cancer, the team is like composed of at least 10 people, and they're all different doctors in different domains, and they're all taking care to make sure that the treatment they're giving you is not going to actually make your cancer worse, okay? And there's a lot of cases where they give radiation, and the radiation makes actually the cancer worse because the cancer changes, mutates, and becomes more resistant to whatever you're doing, okay? Just a little tangent, chemotherapy also is depending on how much of it you do and all that. No, I'm not saying if a person has cancer, don't do this, follow the doctor. But chemotherapy itself can cause resistance and lead the cancer to grow faster. So it can kill it at first, but then when it comes back, it's much more stronger. And again, within the tangent, chemotherapy actually destroys your immune system. And so chemotherapy will potentially lead to you or the person that has cancer dying from actually infection more than the cancer itself. Okay, so I'm just saying this because I saw radiation and chromosome fragmentation, so I'm talking about that side, but this is not related to us. Just imagine that you have a hybrid cell that was just killed using that radiation, and it has one chromosome in it that is human, and the rest is all rodent genome. And so you have that small genome, that one chromosome that is broken into small pieces. Then what you do is you take this cell and you fuse it with a living rodent cell. And you're going to establish about 200 hybrid cells. And these hybrid cells will contain small pieces of the human chromosome. Okay? So you will take pieces, put them in the different cells, and you'll have something like this, right? And then obviously there will be translocations or even fusing of the chromosomes. And so in each line you'll have a different pattern of human chromosome integrated into rodent chromosome. Some crazy stuff, okay? What you do with this then is you look for the presence of an STS marker. So this is the sequence tagged site that is unique in the genome. And then obviously there's many of them in the genome. And so depending on how they are integrated within the man, I don't know, I have to fix this. Depending on how they are integrated with the rodent chromosome, you can then start looking at patterns, right? Because if you take a look at this and you see that you have two sequence tagged sites, then those two sequence tagged sites are probably within the same chromosome, within the same region, because they were integrated together in the same line. Okay? If you find one sequence tag that is in this line and the other one that is in here, that you know that they're probably far from each other, or they're on different chromosomes completely. Okay? In this case, you're looking at one chromosome only. You have 200 hybrid lines, and so these sequence tags are tagged sites are at multiple occurrences along that chromosome, and depending on which ones go together, they will be closer to each other because the fragments will carry, let's say, two sequence tags, and so these sequence tags are close to each other. Okay? Then just by PCR, you can see that marker A is found in line 2, line 1 and line 2. Marker B, line 1 and line 2, so probably marker A and B are close to each other. Marker C found in line 1, line 3, and then marker D is found in line 1, so then marker C and D are probably close to each other, or at least, wait, let me just read this properly. A and B are close to each other. We figure out C from, what am I doing here? Anybody has an idea? A and B, so they're line 1. Actually, you have to read them like, okay, I'll come back to this, okay. C, A, B, C, D. Okay, if you're looking at line 1 and you have A, B, and C, you can figure out that they're very close to each other because you find them there. D, you find it also, so it's probably close to each other. The thing that put me off is the fact that it's just, inserted in another of the rodent chromosome. Okay, that's where I messed up. Is that clear? Make sense? Okay, I think I just said this. The only thing I did not mention is the way that you make these distances. So what you're actually looking at when you're talking about physical distance is the frequency of the markers not being detected in the same cell. And this is just basic math. So the range goes from 0 to 0.5. If you get this number to be 0, then the two markers are always together. If you have 0.5, that means they associate randomly and probably very far from each other. Okay. Good. So the first map that was created was in 1995. It contained only 15,000 sequence tagged sites, and the distance between each site was approximately 200 kb. So you can imagine that some of these are actually longer than big parts of the genome. So this is why of chromosomes. So this is why we call this a low resolution method. Later on, there was an upgrade of this map using genes, and then this was done in 1998. And this map was representing actually 30,000 genes, totally wrong. And it had about 40,000 sequence tag sites. So these are all unique from each other. Okay. And this is the map. Okay, you can take a look at this. So this is the map for all of the chromosomes, and these are all short sequence tagged sites. Finished very fast. Wrapping. No questions. You guys have any questions for me? Anything you don't understand? No? Okay. Well, yep. Sorry?
How did they jump
From
from 15,000 to
15,000 to 40,000, it's just your ability to kind of find different unique sequences within the genome, right? So you need a higher computational power and things like this, okay? And obviously it gets better with time. Within this time, well, it was Windows 98, so I guess it was still bad, but you know, there's just change in like 4 years, 3 years allowed for more computational power to figure all this out. Okay? Yeah?
when we were talking about microsatellites,
Yeah.
there was a slide about hypermarimal mini satellites.
Yeah.
Is this the same thing? Or are we talking about two different things?
So here, they're not the same. Yeah, they are the same things. These are similar things, okay? So mini satellites and microsatellites are also just repeats, right? It's just, I think it's the size of them that is different, right? The microsatellites are the ones that are being used currently, and this is the old way of doing it, okay? They're still repeats. The number of them is highly variable, so they're polymorphic, so that's why they were used. That's just, this method is not used because it required these long gels to be made, right? So now instead what you do is you use these microsatellites using the primers, PCR, and then run a much simpler gel. You can see the difference between this gel and, and this one here, right? These are much more, yeah, let's just leave it at that, okay? Do you guys have any questions about the lecture? The assignment?
Is it just like a summary, or like what do you expect us to do?
For the essay? For the essay, you have to review the paper, basically. So talk about the theory behind, sorry?
A literature review?
Not a literature review. I'm not expecting you to find me 10 papers and then discuss those 10 papers on that topic. What I mean by review is a peer review. So you're reading the paper, you're trying to tell me if the methods they used make sense, what they used as method, are their statistics good, do they have limitations, what are their findings, do their findings contradict each other, are they the same, do they, you know, things like this. So I took a Nature paper, so those ones usually are already peer reviewed. So if you don't find anything from the get-go, think a little bit more, you can, you know, ask me questions for it. But I'm not expecting you to just, you know, review it as if you were like the person that's publishing that paper, okay? It's just the thinking behind it, okay? So you can see the grading scheme that I put in there. Just follow that. If you follow that, you should be okay. Obviously, I'm sure that some of you will start sending me emails to meet up to talk about this. That's perfect for me. You can even make a plan and then show it to me and then go from there. I'm okay with that. Or just send it to me by email. That's going to be a bit easier. Okay. Yes, Julian.
I picked a different article from the Cell magazine, a really recent one actually, about the
Sorry, could you repeat that, please?
I found my own study on the Cell magazine, which was
Yeah.
doing testing samples on the Wuhan market. And
Yeah.
compared that with the lineages of some of the first infected patients in Wuhan. And I was wondering if it's okay to use this study, or if you really want us to do the nature one.
That's a very good question. What do we do? Do you guys want to choose your own articles? So the articles, you will be choosing your own articles in the presentations. The reason why I want to select these articles specifically, it's because I want to give you my side, and then you give me your side, and then we talk about the same topic, so you understand a little bit how the process is done. So if we can, for this one, actually follow that article, and then for the presentations, then we can choose our own articles. Does that?
So the homework that's due today has to be rewritten.
Sorry?
What's different assignment?
Yeah. No, I was talking about the essay that we have to write. Okay, so let me, okay, so we have an essay to write. We have 2 articles to write during this semester. Okay, I see where the mix-up is happening. So the first essay is to be submitted, so I just assigned it. It's to be submitted on the 11th October. Has to be about 5 pages long, and it's on this specific article here. Okay, makes sense? And then that's the lecture right before the reading break. And then when you come from the reading break, I will assign to you another article, and you'll do the same process. Okay? You try to write in scientific style using references that are peer-reviewed, not like the journal stuff we've been doing. Now it's a bit more serious. So first part, start by reading this. And obviously, some of stuff here are quite complex to understand. So even for me, I have to read many times to get the idea. So I suggest that you read it multiple times. Just a little bit of tricks. What I usually do is you don't read it in the order that it's written, right? So read the introduction, go directly to the discussion, right? This way you know what the story is. And then you can get into the specifics of the techniques that you didn't understand and the specifics of the results, okay? Instead of, reading it in one go, you will get tired the moment you get to the methods, right? Because the methods are, very complex to understand. Cool? Good. So have a good weekend. I'll upload the videos tonight or tomorrow.